admitted as many people as possible. So now I think we can start. So the placenta, it is a primary site of nutrients and gas exchange between the mother and the fetus. So it is a primary site of nutrients and gas exchange between the mother and the fetus. So the fetus or the embryo now becoming a fetus into the fetal period, it has grown. So now that's why there is a separate. We are now forming the blood vessels and the heart, so that there will now be sufficient nutrients going around into the embryo or the fetus. So that's why the placenta now comes in. The yolk sac is depleted, the nutrients there is not sufficient. And also the embryo is growing bigger and bigger by the day. So now the placenta has to develop or has to be formed. So this placenta, it's called the fetal maternal organ. It's called the fetal maternal organ because it has got two components. So one component is contributed from the fetus, and the other component actually is also contributed from the mother. So that's why it's called the fetal maternal organ. So fetal part, it develops from the chorionic sac. So remember, in the second week, the chorionic sac was formed, and then the maternal part is derived from the endometrium. So remember, in the first week, I said immediately implantation starts, the endometrium, the name will just change to decidual. So the placenta is the site of nutrient and gas exchange between the mother and the fetus, and it's called a fetal maternal organ because it receives contribution from both the mother and the fetus. Let's describe the placenta. It is a circular and disc-shaped organ, so it weighs about 500 grams. And then it has got two surfaces, two structural components. So we'll describe how each surface on either side, on the maternal side and also on the fetal side, looks like, and then the two structural components, how they are formed on either sides, the mother and also on the fetus. So let's start with the maternal surface. How does it look like? So when we look at this is the maternal surface. So the maternal surface is actually irregular, as you can see. It's irregular, and then it's divided into small lobules. So this is one lobule, another lobule, another lobule. So there are about 15 to 20 lobules. So these lobules are called maternal cotyledons. So these are called maternal cotyledons. The maternal surface is lobulated. It's got small lobules, which are 15 to 20 in number, and these are called maternal cotyledons. Now let's look at the fetal surface. When we look at the fetal surface, you can see it's very smooth because it's covered with amnion. It's covered with amnion, and there's the umbilical cord. So this is the umbilical cord. It's attached to the center of the placenta. Sometimes just here in the middle of the center, at times just slightly away from the center. So it's very smooth and it is covered with amnion, and there's the umbilical cord attached on the center. So these are the surfaces, the maternal surface and the fetal surface. Now we'll look at the structural components, Let's look at the structural components. So this is just showing you how the connection looks like. So this is the fetus, and then the umbilical cord, and this is the placenta. So this is the fetal side, and the other side is the maternal side. So exchange of nutrients and gas takes place in this organ, this fetal maternal organ. So we'll start with the structural component, the maternal component. So the maternal component is derived from the decidual basalis, or what we call the decidual plate. Then the fetal component is derived from the chorion frondosum, or what we call the chorionic plate. So now we'll start with the maternal structural component called the decidual basalis. And then when we are done, we'll look at the fetal structural component, which is called the chorion frondosum. The chorion frondosum. Okay. So we'll start with the maternal structural component, which is called the decidual basalis. Decidual. Immediately the blastocyst started the process of implantation in the first week. The name of the uterine endometrium just changed to decidual. So the decidual separates and fall off during childbirth. So that is the maternal part of the placenta. It will just detach, and then it will fall off, and it will also come out. So the time implantation is taking place, there's a time that implantation is taking place. When it is taking place, we are in the secretory phase of the menstrual cycle. We are in the secretory phase of the menstrual cycle. So during that time, the endometrium is very thick and edematous, meaning it contains a lot of fluids. And the endometrial glands or uterine glands, they are highly elongated and tortuous, coiled, and then they are filled with a lot of glycogen and lipids. So they have just enlarged and elongated. So during that time, that is when implantation is taking place. And we know that that time, where is the nutrition for that for the conceptus? Where is it going to be derived from? From the endometrium itself, the endometrial glands. So they are going to provide nutrition to the implanting blastocyst. And it is also thick due to high levels of progesterone and human chorionic gonadotropin, which is being secreted by the syncytial trophoblast. So the human chorionic gonadotropin mimics the luteinizing hormone. So it will stimulate the corpus luteum of pregnancy to release or secrete a lot of progesterone, because we need the progesterone to sustain this pregnancy and also to enhance development of this conceptus. So the endometrium is actually edematous. So the cells are all filled in with fluids. So this is what we call the decidual reaction. The decidual reaction. So as implantation is taking place, the endometrium is highly edematous. The endometrial glands are tortuous, and the contents increase, and also the blood vessels of the endometrium increase in number. So that reaction, the other reason for that reaction when implantation is taking place, you know, the syncytial trophoblast is eroding the endometrium. So to protect itself from further erosion, that's why the endometrium undergoes the decidual reaction, and also to provide nutrition to the developing embryo. So immediately implantation takes place, the endometrium is just called the decidual. So there will be the decidual will be given different names. Different parts of the decidual will be given different names relative to the position of the blastocyst. So here we can see we have done a horizontal section of the uterus. So this one shaded in purple, this is the decidual, and this is the endometrial cavity. So this is the myometrium. And this we can see is the implanted conceptus, which is the blastocyst. Now, there are three regions of the decidual. So they are given names relative to the position of the blastocyst. So we have this part of the decidual, which is deep to the blastocyst. So this part of the decidual deep to the blastocyst. So this is the part which is going to contribute to the formation of the placenta. So this is called the decidual basalis. This is called the decidual basalis. And the maternal, we have a lot of spiral arteries here. So when the syncytial trophoblast is eroding the decidual for implantation, some of these spiral arteries will be eroded, and they'll open up and they'll fill the lacunae with blood. So there will be lacunae which will develop in the syncytial trophoblast. So as the erosion is taking place, these spiral arteries which have been eroded, so they open up and fill up those lacunae with blood. So the decidual basalis, this is the part of the decidual which contributes to the development of the placenta. So this is the maternal structural component of the placenta. So the maternal part comes from the decidual basalis. And then we have that other part. So that other part which actually surrounds the conceptus, the blastocyst, like a capsule. So this part. So this one is called the decidual capsularis. Decidual capsularis. And then we have that part of the decidual which just lines the other part of the uterine cavity, and it is further away from the conceptus. So this part is called the decidual parietalis. So we have the decidual basalis. This is the part, this structural component from the maternal part, which contributes to the placenta. It's called the decidual basalis. the one which covers the conceptus, the decidua capsularis, and then the part of the decidua which is further away from the conceptus, so just lines the rest of the cavity, uterine cavity. It is called the decidua parietalis. So these are the three parts. So what happens now to the decidua? So as the conceptus, this is enlarging, becoming bigger and bigger. So it will start now moving and covering, projecting into the uterine cavity. So by the time the pregnancy is about 3 months old, this would have, and this capsularis and the decidua parietalis will just meet, and the uterine, the conceptus is growing bigger, and is growing, as it's growing bigger, it's projecting into the cavity. By the time it's 3 months, these two, the decidua capsularis, decidua parietalis would have just been covered up, and it obliterates. Now let's look at the part, structural component, or the part which is contributed by the fetus. So the part contributed by the fetus is called the chorion, or chorion frondosum. So it's called the chorion. So what is the chorion? So the chorion, it is the cellular outermost extra-embryonic membrane composed of the trophoblast, lined with the extra-embryonic somatopleric mesoderm. So chorionic, how is the chorion formed? Formation of the chorion. Remember in the second week. So in the second week, we saw the extra-embryonic mesoderm coming to lie in between the trophoblast and the bilaminar embryonic disc. So it came to lie here. So we had trophoblastic cells coming to fill up the space between the bilaminar embryonic disc and the trophoblast. So this extra embryonic mesoderm was here. And then a cavity appeared which split the extra embryonic mesoderm into two. So that cavity was called the extra embryonic cavity. So this is the cavity which was created, and then it split the extra embryonic mesoderm into two. So there was this part of the mesoderm, extra embryonic mesoderm, which was just solely lining the yolk sac. So that part which was lining the yolk sac, we said it was the splanchnopleric extra embryonic mesoderm. And there was that part of the extra embryonic mesoderm which lined the amniotic cavity, and it also lined the inside of the cytotrophoblast. So this layer of the extra embryonic mesoderm, we said it was called the somatopleric extra embryonic mesoderm. So don't forget the yolk sac, the extra embryonic mesoderm lining it, we said was the splanchnopleric extra embryonic mesoderm. And then the one which was lining the amniotic cavity and then the internal surface of the cytotrophoblast, we said it was the somatopleric extra embryonic mesoderm. Now the chorion has been formed, so it consists of these three layers of cells. So the chorion has the somatopleric extraembryonic mesoderm, and then the cytotrophoblast and the syncytiotrophoblast. So these three form the chorion. So the extraembryonic somatic mesoderm and the two layers of the trophoblast, cytotrophoblast and syncytiotrophoblast, all form the chorion, what we call the chorion. And then we have that part of the extraembryonic mesoderm where the cavity did not extend into. So that part is called the connecting stalk. So the connecting stalk actually just connects the bilaminar embryonic disc to the chorion. It connects the connecting stalk connects, that's why it's even called the connecting stalk. It connects the bilaminar embryonic disc to the chorion, and the chorion is made up of three layers: the somatopleric extraembryonic mesoderm, the cytotrophoblast, and then the syncytiotrophoblast should be here. So that is how the chorion was formed. So this is just explaining how the embryo looks like. So this is just the amniotic sac, blah, blah. So this is just showing you that we have the amniotic cavity here, the epiblast, the hypoblast, and the yolk sac. So we know this is a bilaminar embryonic disc. So this is just the information which is here. So chorionic villi will start to develop from the chorion and cover the whole circumference of the chorionic sac. So that begins in the 8th week. And then later they will become vascularized by the allantoic vessels. Remember in the 3rd week, we said on day number 16, 3rd week, we said the allantois was formed, and it was a dorsodiverticulum from the yolk sac into the connecting stalk. And then from there now, we see the formation of some of the blood vessels. So chorionic villi, what does the word villi mean? Definitely it just has to do with like finger-like projection. Now these finger-like projection are arising from the chorion. So that's why they are called chorionic villi. So finger-like extensions will start developing from the chorion. So these begin in the 8th week, but then we saw some of them starting slowly in the 2nd week. So we'll get to know that. So this finger-like projection will actually develop the whole circumference, that's around the whole circumference of the chorion, projecting into the decidua. So the whole circumference, they'll start projecting out from the chorionic sac, and they will project into the decidua. So let's look at the types. So we have, you can see they have all projected around the chorion, and then they are projecting into the decidua. So some are projected into the decidua capsularis. Some have also projected into the decidua basalis. But then these which are related to the decidua capsularis, these which are related to the decidua capsularis, these are transitory. They will not last long. As the embryo is growing bigger and bigger, they will be compressed, and then the blood supply will just diminish, and then they will just disappear, degenerate, and disappear. So because they're just transitory, so these are called chorion naive. So these are called chorion naive. So these, they will get compressed, and then the blood supply reduces, and then they will just regress. I think by the 3rd month of pregnancy, they will just regress and disappear. So these are called chorion naive. These are transitory, they disappear. So meaning we will just remain with these which project into the decidua basalis. So these which remain and project into the decidua basalis, these are called chorion frondosum. These are called chorion frondosum, and these are the ones which contributes the fetal part of the placenta. So this one which remain to the decidua basalis here. These chorionic villi are the one which contributes to the fetal part of the placenta. And then the maternal part is contributed by the decidua basalis. So this is our decidua basalis here. So 2 types of chorionic villi appear. Transitory ones, chorion naive, and the one which persist and contribute to the fetal part of the placenta. Those are called chorion frondosum. Now let's see the stages in the formation of these chorionic villi. So structural components of the chorionic villus differs at different periods of embryonic development. So we have got 3 types of chorionic villi which appear at different periods of development. So remember in the second week, we talked about the primary chorionic villi. So we said the cytotrophoblastic cells proliferated and projected, formed finger-like extensions which projected into the syncytial trophoblast. So it was more or less like a pyramid extending from the cytotrophoblast into the syncytial trophoblast. So the primary chorionic villi consisted of a central core of the cytotrophoblast covered by a layer of syncytial trophoblast. So these chorionic villi, as they are being formed, they are separated by spaces. Remember those spaces which were formed in the syncytial trophoblast. Those spaces we were calling lacunae, and they were being filled with maternal blood. So when a primary chorionic villi is formed, there will be a space, and then another one is formed, there will be a space. So between each intervening, between each primary chorionic villi, there is an intervening lacunae or intervillar space which is filled with maternal blood. Then development proceeds. So next stage of development, it will be called a secondary chorionic villi. So in the core of the cytotrophoblast, we'll now see somatopleric extraembryonic mesoderm just growing into the core of the cytotrophoblast. And then now there will be 3 layers: outer syncytial trophoblast, intermediate cytotrophoblast, and then the innermost layer is called the somatopleric extraembryonic mesoderm. So now this is just a transverse section. We want to see what layers are found in the different chorionic villi. So primary chorionic villus you find in the middle or the core cytotrophoblast, and then externally there will be a syncytial trophoblast. The secondary chorionic villus, so middle part will consist of the somatopleric extraembryonic mesoderm. Then a middle layer will just be cytotrophoblast, and then the most external layer will be the syncytial trophoblast. Then when it becomes a tertiary villi, it will have all the 3 layers of cells, but then blood vessels will develop inside the extra embryonic mesoderm. So there will just be blood vessels which develop. Then it will be called a tertiary villi. So these are just projections. So I think I have a picture here, don't I? Yes, this one. As you can see, these are the villi which have developed. These are the chorionic villi, and then you can see they are extending into the decidual. So between the chorionic villi, there is this space which was a lacunae. Now it's called an intervillous space, and it's filled with maternal blood. And then the fetal blood is just flowing inside these blood vessels which have been formed inside the core of these chorionic villi. So there is a blood vessel. Then next will just be the extraembryonic mesoderm, and then here we have cytotrophoblast. So this one shaded in very light brown. This is the syncytial trophoblast. So this is a chorionic villus. One chorionic villus, another one, and they are projecting into the decidual basalis. That's the maternal part of the placenta. Now let's see how these villuses are formed. Maybe we can even have a better understanding. So we have this, maybe it's a trilaminar embryonic disc by this time. This is our connecting stalk. And this is the extraembryonic mesoderm. I mean this one here. And then this is the cavity. So cavity, extraembryonic mesoderm covering the trilaminar embryonic disc and lining the internal surface of the cytotrophoblast. This is the syncytial trophoblast, this one in purple with a dotted yellow. And then this lightly shaded part, this is the decidual basalis. So that is the decidual basalis. Maybe I just go through again. So this is our trilaminar embryonic disc. This is the connecting stalk. And then surrounding the bilaminar or trilaminar embryonic disc, we have this cavity. This cavity split the extraembryonic mesoderm into two. So well, this part of the extraembryonic mesoderm, which was covering the yolk sac, so that part is called the splanchnopleric extraembryonic mesoderm. And the part which was covering the amniotic cavity as well as the inner surface of the cytotrophoblast. So this one is called the somatopleric extraembryonic mesoderm. And then this layer, this one which is darkly purple, is the cytotrophoblast. And then this one is the syncytial trophoblast. And here, this conceptus, this whole conceptus, is implanted in the endometrium, which is now called the decidua. So let's see how these chorionic villi are formed. So in the syncytial trophoblast, spaces were formed. So these spaces are called the lacunae. So this is one lacunae, another lacunae here, another lacunae. So as the syncytial trophoblast is eroding and implanting, further implanting and eroding the decidual, so some spiral arteries will be eroded and then they'll just open up. They'll fill up these spaces with blood. So these spaces, the lacunae, they are found in the syncytial trophoblast. So space, so there will be a space. This is a lacunae. This is a space. a lacunae, a space, a lacunae, a space. So this space of the syncytial blast, so this is just this is all, the whole lot is the syncytial trophoblast. Now in the syncytial trophoblast, that is where these spaces, the lacunae, are being formed. So they are leaving a space in between these lacunae. So here we can see. So these spaces are called the trabeculi. They are called the trabeculi. It's a syncytial trophoblast, but now we'll give it a name because it lies between the two lacunae spaces. So it's called the trabeculi. So one trabeculi, another trabeculi, another one, just like that. And then here we have our cytotrophoblast. This is this space here, the cytotrophoblast. Now we have created the trabeculi and we have our cytotrophoblast here. So I think this is just the same information here, which I was explaining using that diagram. So next, we have those spaces. So what is going to happen now? So what is going to happen now is the cytotrophoblast will start to proliferate, the cells will proliferate and they will start projecting into the trabeculi here. They will start projecting into these trabeculi. So when they project into the trabeculi, what are we going to form? We are going to form the primary villi. We are going to form the primary villi. We are going to form the primary villi. So each trabeculus is made up of a syncytial trophoblast and surrounded by a lacunae. So this is the trabeculi and this is the lacunae surrounded by the, so now they are radially arranged. So they are now radially arranged. So now formation of the primary chorionic villi. So cells of the cytotrophoblast will multiply and will start growing into each trabeculus. So the trabeculus has a central core of cytotrophoblast and then it's covered with syncytial trophoblast and surrounded by the maternal blood filling up the lacunar space. It was a lacunar space, but because of the formation of the primary villus, the lacunar space will now be called the intervillus space. So this is what happens, as you can see. Cytotrophoblast will now start projecting and growing into the syncytial trophoblast. So now we have formed these our primary villus because now we have cytotrophoblast projecting into the syncytial trophoblast. Now formation of the secondary chorionic villi. So what happens there now? You can see here, this layer, the somatopleric layer, will also start growing into the central core of the cytotrophoblast. So we have now 3 layers. So this one is the syncytial trophoblast, cytotrophoblast. Now the somatopleric also projects inside. So now it's made up of 3, the villus is now made up of 3 layers. So it's now called the secondary chorionic villi. Now formation of the tertiary chorionic villi. So just within the mesoderm, extraembryonic mesoderm, blood vessels will develop. So when these blood vessels develop inside, then it's called a tertiary chorionic villi. And then blood vessels of the villus will now connect to the secretory system of the embryo. And then fetal blood circulates within the villi. Fetal blood will circulate in the villus. So fetal blood will circulate in the villus, and then the maternal blood will also circulate in the intervillus space. Intervillus space, chorionic villi. Intervillus space, chorionic villi, and these are projecting into the decidua basalis. So now we have formed these finger-like extensions from the chorionic sac into the decidua basalis. So at the end of it, they will look like this. So this is one villus, another villus, another villus, another villus. And then they're all surrounded by these intervillus spaces. And then this is our decidua basalis, or that can just say the decidua. And then these are the blood vessels they have opened up. So they are filling up these spaces with maternal blood. And then this is our embryo. And then this is the umbilical cord. So these villus blood vessels, they will come now to connect to the embryo via the umbilical vein and umbilical arteries into the interior of the embryo. Then this is our placenta. It has been formed. So the fetal part is contributed by these chorionic villi, and the maternal part is contributed by the decidual basalis. So decidual basalis is giving us the blood. We have these blood vessels which have been opened up, and they're filling up these intervillus spaces with maternal blood, but then the fetal blood is just circulating inside these chorionic villus or villi, because there are many. Now that we have formed these villi, so the most external layer is the syncytial trophoblast. The most external layer is the syncytial trophoblast. So we have the syncytial trophoblast externally, then deep to that we have the cytotrophoblast, and then again deep to the cytotrophoblast we have the somatopleric extraembryonic mesoderm. So that is the arrangement. But then the cytotrophoblast will grow and extend outside, will extend externally and cut off the syncytial trophoblast. So the cells will proliferate, extend outside the villus, and then they will create a layer, which will be now the most superficial layer. So this layer is the one which is going to come into contact with the decidua basalis. And this will be and then cover up the syncytial trophoblast, because the syncytial trophoblast previously was the most external layer. But then the cytotrophoblast will grow, extend outwards, and then form a layer separating the decidua and the chorionic villi. So that layer of cells is called the cytotrophoblastic shell. Why is it formed? For me, I think it's because the syncytial trophoblast, they have got that property of eroding, just keep on eroding. So now that we have the placenta being formed, do we need the syncytial trophoblast to keep on eroding? No. So that's why these cells had to come out, proliferate, and form the outermost boundary. So separating the syncytial trophoblast from the decidua basalis. So this new layer which has been formed is called the cytotrophoblastic shell. So the cells of this shell will multiply, and the placenta now will increase in size. So this is now the new arrangement. So as you can see from here, so these cytotrophoblastic cells emerged, extended externally, and formed the outermost layer covering the syncytial trophoblast. So the cytotrophoblastic shell is the externalmost boundary of the chorionic villi, separating it from the decidual basalis. So now we have formed our placenta. So the placenta has been formed. From the fetal side, we have these chorionic villi, and from the maternal side, we have the decidual basalis. So fetal blood is circulating in the vessels which are found in the chorionic villi, and maternal blood is actually circulating in the intervillous spaces in this. They were formed in the syncytial trophoblast. And from the maternal side, we have the decidual basalis, and we have these spiral arteries which were eroded, and they keep on filling the intervillous spaces with maternal blood. So this is our placenta, which has been formed. This is how the placenta.
Just a question. The placenta is surrounding the embryo right now.
No, remember there was those which died. So there were these, just showing you how they were formed. But there was this one which were related to the capsularis. What happened to them?
We said the chorionic villi
Yes.
died.
Yes. So these are the ones here. They died. So it's just showing you how they are formed. All of them, they were formed. But then the concept should be these ones should die. So we are going to remain with only these ones here, which are related to the decidual basalis. Are we clear?
Yes, we are clear. Thank you.
Yeah. So they were all formed. Oh, I said they were all formed all around the whole circumference of the conceptus. But those which were related, so these ones should be these ones here, to the decidual capsularis. These are the ones which regressed, and then we just remained with these ones which are related to the decidual basalis. So we're just showing you how it was formed. So frondosome, these are the chorion frondosome which remained. These are the naive which disappeared. Okay, now we have gotten the concept. So trophoblastic shell has been formed. Now each villus is formed. So when it is formed, it's first called the anchoring villi. So the villi which are first formed, they are attached on the fetal side to the extra embryonic mesoderm. And then on the maternal side, they are attached to the cytotrophoblastic shell. And they are called an anchoring villi when they are just formed. So when they are formed, they look like this. So they are called an anchoring villi. You can see it's attached to the extra embryonic, somatopleric extraembryonic mesoderm. And then here it is attached to the cytotrophoblastic shell. As you can see, it's lightly shaded here. Though if you've got eyes, you will be able to see there are two blues here. So this is the cytotrophoblastic shell. So this is the first one which is formed. When they are formed, they just look like this, anchoring villi, and then you can see these blood vessels. Yeah, so these are the blood vessels, and then they are all surrounded with this, what we call intervillous space. So this is an intervillous space which is surrounding it. Yeah. And then it will start now giving out branches. It will start now giving out branches. So each anchoring villus will
so
now...
Madam, query on the previous diagram. We said that the mother's blood is all in those blue spaces we call the intervillous space.
Now they'll start surrounding, because these have now formed. So now they'll just surround the villus. They can't mix. You understand? So yes, maybe I use it's understood. So you can see these are the intervillous space. That is where the maternal blood. You see it's surrounding this villi. This is the villi. It's all surrounded. Even here, it's all surrounded. Then when it slims down, maybe it trims down, it comes like, it starts looking like this. So meaning it will now be surrounded by the intervillous space. So all this is maternal blood. Is it clear? intervilus space. So these are the intervilus spaces. Intervilus spaces. This is all blood. This is all blood. This is then the green part. If you can notice, the green part is the decidua. So this is just giving us the arteries are coming from there, and then they are filling these spaces. Remember, those spaces were created in the syncytial trophoblast. Are you clear? Yes, I need the feedback from you. Are you clear? The person who asked. Are you with me? So this is the fetal part. This whole shaded blue is the fetal part. And these are the viruses which were formed from the chorion. So these are the viruses, the finger-like projection. And then this is the decidua basalis now, the one shaded in green. So the intervilus spaces, they were formed in the syncytial trophoblast. So this should be the fetal part. Syncytial trophoblast was coming from the fetal part. So now that they have slimmed down, they have grown. So it means now they are all projecting into the syncytial trophoblast. So if you can see it from here. So this is the villi which has been created. It just elongates. So meaning it will just be surrounded by the intervilus spaces with blood. Okay. So now each anchoring villus will consist of a stem. So the truncus cori. So it was just first an anchoring villi, and then it will have a stem. So the stem is called the truncus cori. So further development of the chorionic villi. So it will give out branches. So these branches are the ramus cori. Then it will give out very small branches. So these smaller branches will grow. These are called ramuli cori. And then they will just even be further small, these other smaller branches. These are called floating villi. So the ramifications, these are just to increase the surface area, to increase the contact with the maternal blood. So maternal blood and the fetal blood, they will never mix. So the maternal...
madam, what is the difference between the fetal cotyledon and the intervilus space?
I will come to that. It's coming to be explained.
All right.
So let me just first know how these are branching. So this is a chorionic villi now with its branches, ramifications are being formed. So to have, this is now a fully mature chorionic villi. So it has a truncus cori, then a ramus cori, and then these smaller branches are called ramuli cori, and then these are the floating villi. So this is just to increase the surface area or the contact with the maternal blood. So these are just floating and they are swimming in the maternal blood. But then the fetal blood is secured in the villi, in these villi. So all these, they all have the blood vessel. So this whole villi is called the fetal cotyledon. Even made me to start talking about the fetal cotyledon. So this whole villi is called a fetal cotyledon. And then this is the decidua basalis. So it will start sending in septa. So dividing the decidua basalis into what I mentioned earlier on, the irregular lobulated surface of the maternal side. So forming the maternal cotyledon. So these septa which are being formed here. So this is one maternal cotyledon, and this is also another maternal cotyledon. This septa grows inward, so this other one, so this forms another maternal cotyledon. So it's just the decidua basalis sending in septa into the placenta. so forming the lobulated part, irregular surface of the maternal side. But also from the fetal side, this chorionic sac, when these chorionic villi were being formed, so these are the same fetal cotyledons. So in a one maternal cotyledon, you can even find as many as 4 fetal cotyledon. So in this one maternal cotyledon, you can find 4 fetal cotyledons. So the fetal cotyledons are just these same chorionic villi. Okay, are we together? So I just hope we are moving together. So now, I think this one has been explained. So it's just now trying to enlighten you that the placenta now has developed. So there are 2 parts, the fetal and the maternal. And then there are 2 surfaces, fetal and maternal. And then there are 2 types of cotyledons, which I've just mentioned. The fetal cotyledon, which is just the chorionic villi itself, and then the maternal cotyledon, that is just a lobule, which has been formed from the decidua basalis. So all that has been explained, so I don't need to go through it again. So after birth, the placenta always has to be inspected for missing maternal cotyledons. So if some of the maternal cotyledons, they remain still attached, so that is what causes bleeding after delivery. So there are some women who have died because of severe bleeding. It's because some of the cotyledons did not fully detach from the uterine wall after birth. So everything, the whole of the placenta has to move, has to detach and come out. So if some of them still remain attached, then there's a problem. We are going to have maternal bleeding. The mother will bleed. So this is just another diagram, just illustrating the placenta. So this is the decidua basalis, this one in green, and you can see these are the blood vessels. So they send in septa, then forming lobules from those you were seeing from the maternal sides because of this extension of septa into the fetal side. So we have one maternal cotyledon, and then we have these blood vessels. They are filling these spaces, which are colored blue with blood. So these are the intervillous spaces. And then these are the chorionic villi. So this is a fetal cotyledon. So in one maternal cotyledon, you can even find maybe as many as 4 fetal cotyledons. So blood is trickling in into the intervillous spaces after consumption and exchange of nutrients will diffuse into the blood vessels of the fetus and then into these chorionic blood vessels, and from there into the left umbilical vein, and then into the embryo. Nutrients are being taken into the embryo. So this is the fetal circulation. Then this is where the exchange of nutrients and gases are taking place. This organ called the placenta, and then this is being transferred into the embryo through the fetal circulation. So this is the placenta. So when the placenta, when viewed from the maternal side, these are the lobules you are seeing. It's because the decidua basalis extends septa inward. So these are the lobules you are seeing. It's because of the extension of septa into the fetal side of the placenta. So these are the grooves you see. And then we form the maternal cotyledon, maternal cotyledon. Then the fetal side also, It's covered with amnion. We saw that it was covered with amnion. And then we have the umbilical cord attached at the center. So this is our umbilical cord here. Umbilical cord, and the arteries at the center. So here it's covered with amnion. And the fetal side is contributed by the chorionic frondosome, seen as the plate called the chorionic plate. Then from the chorionic plate, we have 40 to 60 extensions. That is from the chorion frondosome. So these are called fetal cotyledons. So these same villi are called fetal cotyledons. And then they extend towards the decidua basalis. So fetal cotyledons, and then they extend towards the basalis, decidua basalis. These are the chorion frondosome. So fetal cotyledon consists of a stem villi, truncus chori that show a number of branching. So those branches, which I already showed you, the stem villus, ramus chori, ramuli chori, and then the smaller ones, the floating villi. So these are the fetal cotyledons. So 15 to 20 maternal cotyledons in the placenta. So each maternal cotyledon contains 2 to 4 villi with their branches. So these are the ones I'll keep on repeating, and their branches. So we have formed our placenta. Now the peripheral margins of the placenta. So it presents what we call a fetal membrane. So this fetal membrane is contributed from inside outwards by the decidua, capsularis, parietalis, chorion laive, and the amnion. So these are the peripheral margins of the placenta. And after birth of the child, the placenta is shed off along with the decidua. So these are the peripheral margins. You can see, just showing you the peripheral margins. So the peripheral margins from outwards, inside outwards. So we have the decidua capsularis, this one from inside outside, capsularis, parietalis, chorion laive, and then the amnion. You can see the amnion now from outward inwards, inwards outwards. So these are the peripheral margins. And this is our placenta here, which is confined here. Now the placental membrane barrier. So these are the barriers which separates the maternal blood from the fetal blood. So maternal blood and the fetal blood can never mix. Maternal blood and fetal blood can never mix. They can never mix. So there are these what we call placental barrier or placental membrane. So these membrane are just made up of layers of the wall of the villi. So these are the same layers. So that is where the nutrients and the oxygen will diffuse few to reach to the fetal blood. So let's look at these layers. So the constituent structures forming the placental barrier or the maternal fetal barrier. So these extend from the maternal erythrocyte to the fetal erythrocyte. So these are as follows. So in early pregnancy, the barrier, the innermost, so we are now inside the chorionic villi. So there will be the endothelium of the fetal blood vessels and its basement membrane. So what other layer are we going to find? So we are going to find the extra-embryonic, somatoplasmic extra-embryonic mesoderm, and then we'll come to the cytotrophoblast and its basement membrane, and then the syncytial trophoblast. And then we have the intervillous space where with this maternal blood. So maternal blood has to go through all these layers to reach the nutrients and gas from the maternal blood has to diffuse through these layers to reach the fetal blood. So that's what we mean by placental barrier or maternal fetal barrier. So let's use this diagram. So this is the intervillous space. There is maternal blood. And then this is the chorionic villus. So these are the blood vessels. And in the blood vessels, that is where there is fetal blood. So for nutrients to diffuse, they have to pass through these barriers to reach the blood in these blood vessels. So this is where we say we have the endothelium of the fetal blood vessels, endothelium of the fetal blood vessels and its basement membrane. Then this other layer is the somatoplasmic extra-embryonic mesoderm. This other layer, this one is the cytotrophoblast, and then this other layer is the syncytial trophoblast. And then now this is where the maternal blood. So all this from here to here, that is the placental barrier. These are what we call the placental barrier. So later as pregnancy progresses, so there is a high demand for nutrients. So what is going to happen is we are going to see the disappearance of the cytotrophoblast, as you can see here. And then the syncytial trophoblast will thin. You can see here it from this thick to slimming, to thinning. And then the stroma will become edematous. And then the fetal blood vessels, they were in the middle of the villus. Now they will start moving to the periphery. And then we are going to epithelium of the fetal blood vessels resting on this basement membrane. And then it is only related to the syncytial trophoblast. We have removed the cytotrophoblast because we need as much nutrients and oxygen to diffuse to cater for the demand of the growing embryo or fetus at that time. Now, what is the function of the placenta? Protection, nutrition, respiration, excretion, and also hormone production. I don't need to explain this. So protection from bacteria, harmful substances, nutrition, that's where gaseous nutrients exchange takes place, respiration, gaseous exchange, excretion, waste products will be released there. And then the placenta also produces hormones. So what are the hormones produced by the placenta? So this syncytial trophoblast will actually synthesize proteins and steroid hormones. So the protein hormones, we have the human chorionic gonadotropin. So this one mimics the luteinizing hormone. So it will stimulate the corpus luteum to function and to produce more progesterone. And then the human chorionic somatotropin. So this is an anti-insulin effect on maternal blood. So that will lead to increased levels of glucose in the blood so that can easily diffuse and be utilized by the fetus. Human chorionic thyrotropin. This just increases the rate of metabolism throughout the pregnancy. And then human chorionic corticotropin. So this one will stimulate the pituitary gland to release adrenocorticotropic hormone so that there's increase of fluids in the body. And then the steroid hormones, we have progesterone to maintain the pregnancy, and then estrogen, which will promote uterine growth and development of the mammary glands. And then initially, in the initial stages, the placenta will be storing glycogen, calcium, and iron. So now as the embryo develops, organogenesis takes place, that function now will be taken up by the liver. Then we have different shapes of the placenta, but the normal shape is discoid or disc-like. It's the one which we are from describing. But then there are instances you can find a bidiscodal. So this one consists of two separate discs, so that is away from the normal. Then it's supposed to be round, then it will be oval. You can also find a triangular shape, and then the one which is just irregular. So irregular shape. So which one is the irregular? This one you can see it's irregular in shape. So this is also not the normal. Then you can find one which is lobulated. It's got lobes in it. So the lobulated one here is got lobes. Different, this is another lobe. So this is also another abnormal one. And then we have diffuse placenta membracea. So this one looks like this. You can see a lot of chorionic villi all over. So that's what we call, yeah.
What causes low line in placenta? Could it be due to the like at this point in time when the placenta is fully forming, or could it be because of the weight of the baby?
Low lying
Low line in placenta.
or placenta previa?
Yes.
It's just maybe abnormal implantation. So maybe the implantation took place at a very low level. So that is where the placenta will also form. Remember implantation?
Mm.
Yeah. So if the embryo implanted at a very low level, so the placenta will also start forming there. So that's why we call placenta previa.
That's what also causes spotting.
Spotting?
Yes.
Spotting is caused by a lot of things. So early in pregnancy, before the placenta is formed, yes, spotting is there. Spotting can be there. But after the placenta has formed and the pregnancy has progressed,
Yes.
the spotting is not supposed to be there. So, but if the placenta has implanted very low, then spotting, yes, can be there, but it's not supposed to be there. It's abnormal.
It's
It's
abnormal.
just showing that you're going to lose that pregnancy. So it's just an indication that you could lose that pregnancy. Spotting is not supposed to be there. But if it implants very low, yes, there can be some spotting.
Then what if like after the implantation, then there is low lining and the spotting, then the baby, you get to give birth to a very healthy baby. Could there be any other effects in future, maybe if you were to have another pregnancy?
No. There are people who even have their menses even up to maybe 7 months, but it has got no impact on the baby. We have seen women who are even menstruating. They didn't. If maybe the pregnancy is not even showing, they'll even think they're not pregnant. But then it's not supposed to be there. There's not supposed to be any spotting. But if there's an anomaly, you just go to the hospital and then they'll try to find out the cause of that.
Thank you very much, Doc.
You're welcome. So we can also have placenta saxenturata. So this one is a small part of the placenta. It's just separated from the rest. So this one here. So a small part is just separated from the rest. Then we can also have a fenestrated one, meaning there's a hole, which is not supposed to be there. So this one here, it has a hole here. So I don't know, during development, some anomaly could have occurred. And then we have circumvallate. So in this one, the peripheral edges of the placenta is covered by just a circular fold of the decidua. So it's just supposed to be on one side, but then the whole placenta is covered. So that is what we call circumvallate. So as you can see from here, fold of decidua, which is supposed to, on the fetal side, is not supposed to be there. So this is our placenta. And then we have these fetal extraembryonic membranes. So these ones, we actually discussed them in the 3rd week, 2nd week. So I won't go back. So you just have to visit the notes the 2nd week and 3rd week. So trophoblast, chorion forming, placenta, the chorion. We talked about how we discussed how it was formed. In amnion, we discussed that and saw how it was formed, the yolk sac, primary, secondary, and up to tertiary level. So tertiary was formed last week when we were looking at the 4th week of development, when the primitive gut was formed, and that part which remained outside was the tertiary. Primary, we saw that in the 2nd week, secondary in the 2nd week. Then the allantois, we also discussed that, the connecting stalk and the umbilical cord, we discussed that in the 2nd week, and also in last week we did that. So this is just a link you can follow just maybe to remind yourselves of these fetal extraembryonic membranes. So those are the structures which develop outside the embryo. Then we have, what are their functions then with these same fetal, the extraembryonic membranes, what are their functions? So mainly for protection, respiration, excretion, nutrition. And why are they important? These extraembryonic membranes for performing prenatal diagnostic procedures such as the chorionic villus biopsy, which we discussed last week, and amniocentesis. This one was also discussed last week. Now let's go to twinning and multiple births. So multiple births, this one is just where more than one fetus is carried to term in a single pregnancy. So a mother can give birth to 2 infants at the same time. These are called twins. There are 3 triplets, quadruplets, and even more can happen. So let's look at the types of twinning. So twins can be produced in 2 ways, dizygotic twins. So these ones, the dizygotic twins, are formed when 2 ova are released at the same time, they are shed at the same time. They are released from the ovaries at the same time, and then they are fertilized by 2 different sperms. So we can say they are 2 siblings which are born at the same time, 2 siblings who are born at the same time. So these are fraternal twins. They're actually different. Can be a boy and a girl, but they don't look alike. Or even a girl and a girl, but they don't even look like a boy and a boy, but they don't look alike. Even the genetic makeup is different. So when you look at them during development, so each fetus will have its own chorionic sac, that is the placenta, and it will also lie in its own amniotic cavity because they are 2 separate individuals. So this one twin will have his or her own placenta and lie in their own amniotic cavity, and the other one will also have their own placenta and they'll lie in their own amniotic cavity. So these are the dizygotic twins, and they are more common in human beings than the monozygotic twins. So you can see, so 2 zygotes for the fertilized from 2 different sperms, and then as they progress, they have his own amniotic sac, chorionic sac, which is the placenta, and it's lying in its own amniotic cavity. And then we have monozygotic twins. So these arise from a single fertilized ovum. So they arise from a single fertilized ovum. So their genetic constitution is just the same because they arose from single fertilized. So the the oocyte will be ovulated. This one sperm will come and fertilize it. And then we're going to form a zygote. But from this zygote, maybe the cells will separate and form two separate human beings actually, who have the same genetic constitution. So these, in appearance, they are a carbon replica of each other. So these are the identical twins, what we call identical twins. So it can arise, we can say monozygotic twins are produced in one of the following ways: early blastomere separation. So immediately the zygote is formed. At two-cell stage, the two cells will separate and start, and two individual separate human beings will start developing. So at this stage, we are only at the cell stage. We haven't formed the trophoblast, and we also haven't formed the amniotic cavity. So when they develop, these fetuses are going to have their own placenta. Why? Because we haven't formed the trophoblast yet. So meaning each one will have their own placenta, and they're also going to have their own amniotic cavities. That's where they are going to, or amniotic sac, because at this time we're just at cell stage. No trophoblast has been formed, and the embryoblast has also not yet been formed. So when they start developing, they'll start forming their own trophoblast, their own embryoblast. So these are formed at early blastomere separation. First cell division, when we have two cells, and the two cells will now separate and start forming individual human beings. So at that stage, we're just at the cell stage. We haven't formed the trophoblast. So meaning each of these will form their own trophoblast. And we haven't formed the embryoblast. So each of them will form their own amniotic sac. So they will lie in separate amniotic sacs, and then they'll also have their separate placenta. So the incidence is 25 to 30%. And then we have identical twins, which are formed at the level where now, with the level we reach where we have inner mast cell and the trophoblast around it. So inner mast cell, which is the embryoblast, has been formed, and the trophoblast has also been formed. So these two fetuses have a common chorionic sac. So because of that, the trophoblast has already been formed, the outer cell mass. So meaning they can't form another one. So they are going to share the placenta. But then we haven't reached a stage where the amnion has been formed. So at that point, each one can have their own amniotic sac, so they can lie in their own amniotic cavity. So percentage of these is 70 to 75%. So we have the trophoblast has been formed, and then we formed the embryoblast. So the embryoblast duplicates itself. So development. So because we have already formed the trophoblast is the one which is going to form the placenta. So usually they share the placenta. So at this stage, we haven't formed the amniotic cavity. So each one of these can form their own. So they will lie in their own amniotic cavity, but then they will share the placenta. Because at the stage they were formed, the trophoblast had already been formed. So we can't form another one. So during formation of the placenta, they just have to share the placenta. But at this stage, we haven't formed the amniotic cavity. So each one will form their own amniotic cavity. So they'll lie in their own amniotic cavity, but then they'll share the placenta, because at this stage, we already have the trophoblast. And then duplication of the embryonic disc. So that time, maybe it's a bilaminar embryonic disc. So maybe at that point, it has to separate, form two. So at that time, the trophoblast has already been formed, and at that point we have already formed the amniotic cavity. So we can't form any other. So since they have duplicated, meaning they have to share the placenta and also have to share the amniotic cavity. So these ones, the incidence.
Will the fetus develop inside the placenta or outside the placenta?
Outside.
Okay, madam. Thank you very much.
How can the fetus develop inside the placenta?
Madam, confusion.
No, you understand, I know.
Okay, thank you, madam.
No, you will understand as you go through the notes. Read and read and read.
Okay, madam. Thank you very much.
No, you're welcome. So duplication at the embryonic disc. So this time the trophoblast has already been formed, and maybe we're at the bilamin embryonic disc. So this time around, maybe the amnion has even been formed, but then just duplicates, just separates and forms two different human beings. So these human beings, the fetuses, because we already formed the trophoblast, so they are going to share the chorion, which is the placenta, and then they will develop each one in the common, they will develop in one amniotic cavity. Yeah, so they will develop in one amniotic cavity because the amniotic cavity has already been formed and then it just duplicates. Yeah, so I hope we are good. So this one is very rare, and this one leads to chances of having Siamese twins at this stage. So if the duplication is incomplete, the separation is incomplete, it can lead to Siamese twins at this stage, at the bilaminar embryonic disc stage level. It can lead to Siamese twin. So a clinical correlation, we have placenta previa. So normal attachment of the placenta is in the upper part of the uterine segment. So this attachment of the placenta usually will be formed just at the lowest level near the external os of the cervix, because implantation of the blastocyst was also done at a very low level. So that is what we call placenta previa. So implantation is supposed to be somewhere here, but then maybe implantation took place here. So even the placenta will form here. So it's in types. There's the first degree, this one is just near the os and also the uterine wall. And then the second degree is just almost near the entrance. The placenta is formed just near the entrance. And then there's a third degree, the external os here is closed off. So this is a high risk pregnancy. And then fourth degree, this is the worst, placenta being formed here. Wow. So it can lead to even losing of that pregnancy. Even the mother itself can be affected. So amniotic fluid. So amniotic fluid, 10th to 12th week, 400 milliliters. Then it will increase up to 6 months, and then it will decrease. So at 28th week, 100 mil, normal is 800 milliliters, and then at full term, supposed to be 700 to 1000 milliliters. But then if there's excessive volumes of amniotic fluid, e.g. almost 2000 milliliters, which is abnormal, this is called polyhydramnios. It's called polyhydramnios. So this is abnormal production. So this one can be attributed to atresia of the esophagus. So the esophagus, we're going to look at it when we start looking at systems. The esophagus is actually closed off. There was no recanalization during its formation, so it's closed off. So the fetus is unable to swallow. So ideally, the fetus is supposed to be swallowing that amniotic fluid. But if it's not unable to, then it can lead to this condition called polyhydramnios. Or sometimes even the defect of the central nervous system can also cause that. So CNS prevents swallowing of the amniotic fluid by the fetus, because swallowing is just a process which involves neurons and the brain. And there is also a condition called oligohydramnios. What is oligohydramnios? Scanty amniotic fluid. So below the normal milliliters. So normally 700 to 1000, but then it's somewhere around 400, then it's oligohydramnios. So this causes placental insufficiency with reduced placental blood flow. So maybe the loss of the amniotic fluid was maybe due to preterm rupture of the amnion. And sometimes it's also attributed to renal agenesis. So the kidneys did not develop. So the fluid is also contributed by the urine from the fetus. So fetus urinates into the amniotic cavity. So also contributing to the levels of the amniotic fluid. So if kidneys fail to develop, it can cause oligohydramnios. So because of scanty amniotic fluid, it can lead to difficulties during labor or childbirth. So amniocentesis, I think we discussed it even last week. So it's a technique to collect amniotic fluid for diagnostic purposes. So it's usually done during the 15th to 20th week of pregnancy. So that's why I emphasized last week, it's very important to know the age of the pregnancy. So that can also guide you as to when you can do these procedures. So if you don't know, maybe there's a problem, you're suspecting maybe congenital abnormality, especially these chromosomal abnormalities. Now you want to take some of the cells so that you can do a genetic test somewhere. So you're suspecting a genetic disease, so Down syndrome. So you can just do an amniocentesis, just get the fluid. So there are some cells there which you can also get and take them to confirm if there is a genetic condition. But if you don't know the age and you go and do an amniocentesis, that can also be a danger to that pregnancy. So you can also detect neural tube defects and also a lot of things you can detect when you get a sample from the amniotic fluid, even some of the sex of the fetus and all those. And then chorionic villus biopsy. This one is done early. So you can do this one early before amniocentesis. So this is the same just for diagnostic purposes, but it's done earlier than amniocentesis. So know the age of the pregnancy. And then Siamese twins. So that it was because of incomplete duplication of the disc, the bilaminar disc. So you can have craniopagus joints at the head. So this is craniopagus. Thoracopagus. So these are the ones which are joint here. Pygophagus. These are the ones which are joint at the sacral region. Cephalothoracopagus, fusion at the thorax and the head. And there's also a parasitic twin. So maybe the twins were formed, but one other one, due to lack of blood supply, it failed to thrive. So you find it stuck on top of the viable twin. So you find it stuck on top of the viable twin. So what caused the twins to be thinking the same, even when you examine them in terms of the test, we'll get the same results after separating them. I don't know what you mean. So the twins to be thinking the same, even when you examine them in terms of the test. Yes. If they are monozygotic, then they should be the same result. But if they are fraternal twins, you can find one is Down syndrome, the other one is normal. You can find their twins, one is an albino, the other one is normal. So those can happen. Not always that they have to have the same results, but for these identical ones, because they developed from one egg, the result should be just the same. So we are done for today. Thank you so much for joining. So any contributions, questions? So we are done for today. I hope I will just...
Yes, we do have questions. Just a minute.
Mm-hmm. Is it possible for one twin to swallow the other? No, I'm posing that question back to you so you come and explain to us next week. Yeah. You can go ahead.
So for the fertilization site, we said it was the upper portion of the uterus. Was it anterior or posterior?
So it can occur anywhere. So if it is anterior, those are the women you see the tummy is protruding. Others, you can't even know that they are pregnant because it was somehow posterior. You get the point, eh?
So both are normal, isn't it?
Yes, they are normal. So if the one who's implanted anterior, that's where you see a big belly in front. But the one who's almost implanted at the posterior side there, but not very too high there, you won't even see the pregnancy. So you see the difference. Mm-hmm.
For the type of implantation, we said it was intertissue or interstitial.
Interstitial. It's buried under the endometrium. You can see the decidual capsularis covering it. Did you see that? So that's interstitial implantation.
And then for premature birth, we said it's before how many weeks?
The normal, it's how the pregnancy. How long is one pregnant?
40 weeks.
So if it's before that, then it is premature. But then we'll look at the lung development. We'll discuss that when you start looking at systems. Yeah?
And then for corpus albicans, we said it's the degeneration of the corpus luteum. So if there is a pregnancy, the HCG hormone doesn't allow it to degenerate. So does corpus albicans still form when there is a pregnancy?
No, it won't form, because it will now become the corpus luteum of pregnancy.
So no corpus albicans during pregnancy?
No. So if there's no pregnancy, it will become the corpus luteum of menstruation. Now, because the progesterone levels are going down, to start regressing, to also respond to that. And then it will just become the corpus albicans, a white substance.
And then the difference between follicular and granulosa cells.
Follicular and granulosa cells. So follicular, those are the ones which surround the oogonia. Follicular cells, these are the flat cells which surround the oogonia. Immediately the oogonias enter into they reach the developing ovaries. So there are some cells which surround it, which come from the coelomic epithelium. So that's now in systemic. This is now in systemic embryology. So those are the flat cells which surround it. So now it will change into a primary oocyte, and the oogonia will replicate the DNA, and then it will start meiosis 1. And then it will be halted in prophase diplotin. And then it is surrounded by those flat follicular cells. So those flat follicular cells plus the primary oocyte inside it, that is what we call the primordial follicle. So during ovulation cycle, these follicular cells will do what? Will start developing. So there will be a stage when these follicular cells will proliferate and increase in number. So that's when the name will just change to granulosa cells. Is it clear?
Crystal clear. And then for the caudal plates, we said it's the precaudal or procaudal plate.
Depending on the book, some book will say precaudal, but most books will say procaudal plate. So depending on the book you are using. So procaudal plate is the place, procodo is a place where there's just ectoderm and endoderm. And then there's also a precodo, I think, should be a mesoderm, which will induce the stranger structures to start developing. So if you read in the notes of the 3rd week, you will find that place.
So meaning in the assessments, we can use any, isn't it?
Yes. But use mainly the procodo plate. So that came from, which book is that? Medical, is it Langman's? Yeah, so that one came from Langman's. Procodo plate, please, Langman's embryology.
All right, I think that's it.
Okay, thank you. So any more questions, you are free to consult me, even call me. I will be able to clarify. Especially around lunchtime, if you have my number, if you need something, clarification, you can call me. And if you live somewhere around Levi Mwanawasa University, I'm in the university there, you can still call me, you can come to my office there if you need clarification.
Okay, madam, just one more question. For G0 phase, it was not mentioned. So between interkinesis, do we have G0?
Okay, G0, I didn't mention it deliberately. Why? Because I know there are my colleagues who elaborate more on that. Me, I'm not going more into physiology, you know what I mean? I just wanted to enlighten you. So that's why I didn't mention all those. I know that there's physiology, there's biochem, there's the microbiology. They'll teach you all those into details. So that's why me, it's more of embryology, just to give you an idea of how these are occurring. Yeah, so if you want that, maybe consult physiology textbooks. So me, I didn't go all that way, but it's supposed to be there. So me, I originally avoid it because I'm not teaching physiology. I know my the physiologists who elaborate more on that. Yeah. So that's it. Okay. So number I will share. Thank you so much for listening.